So let's wait another minute maybe before we kick off for more people to join us.
Okay.
And then we'll start. So I see more people are joining. Just maybe we can start 2:02.
And you can see my spider full screen as well. Really well, it's fine. Just checking.
I can see your screen fine. Yeah.
It's on full screen. Is that true?
Yeah, it's full screen mode.
Perfect. Okay.
Zoltan, if you can just speak a little bit louder because it sounds like a filter or something.
All right. Yeah, let me just.
Now it's much better. Yeah, if you're closer maybe to the mic.
Just move the microphone a bit closer, I guess. Yeah. I hope it's better now. Can you hear me better?
much better. Yeah.
Perfect.
Okay, so let's start maybe with the introductions and we give some time to people to join. So hello everyone and welcome to the Institute for Molecular Science and Engineering's webinar with title Quality by Digital Design and Techno-economic Modeling of RNA Vaccine Production for Pandemic Response. Today we've got Dr. Zoltan Kis presenting his work on that front, who is a lecturer at the Department of Chemical and Biological Engineering at the University of Sheffield. So as I give some time to people to join us, maybe I'll start by introducing myself. I'm Dr. Maria Papathanasiou. I'm a lecturer at the Department of Chemical Engineering and IMSE affiliate. And just some helpful sort of links that you can find in our chat function. And those include our IMSE Eventbrite page, where you can find different events run by us, including the annual lecture that will be given by Professor Teresa Lambe, who is principal investigator and group leader at the Jenner Institute in the University of Oxford. Links to the IMSE YouTube channel, the blog, as well as the podcast, Never Lick the Spoon and briefing papers. A few housekeeping rules to make sure we run a smooth webinar today. Just make sure to have your microphone and video switched off. And if you do need to ask a question, you're of course very welcome and encouraged to do so by putting it in the Q&A section of that Zoom meeting. And then we will keep an eye on the questions and I'll ask those to Zoltan myself after the end of his presentation. Please do not put your questions in the chat function. Just use the Q&A option for that. If you do have any technical difficulties today, please message IMSE Imperial privately and a member of staff will try to help you as soon as possible. So going back to our speaker, as I mentioned, Dr. Zoltan Kis is a lecturer at the Department of Chemical and Biological Engineering at the University of Sheffield and a dear colleague and collaborator of mine as well. Zoltan has been developing and modeling rapid response vaccine production platform technologies like the RNA. And this work today is addressing the challenge of producing large volumes of vaccines rapidly at high quality and low cost for pandemic responses. So Zoltan has previously worked as a research associate in the Future Vaccine Manufacturing Hub and the Center of Process Systems Engineering at the Department of Chemical Engineering at Imperial. And he has obtained his PhD in bioengineering from Imperial, and he also holds an MSc in applied biotechnology and a bachelor's degree in chemical and biochemical engineering. So without any further delays, I would like to give the floor to Zoltan, and I'm very much looking to the presentation.
Thank you, Maria, for that wonderful introduction. So I've actually just joined Sheffield last month, and before that I was at Imperial working with Maria and others as well. So the work that I'm going to present to you today was actually done at Imperial College London. This was done in the frame of the Future Vaccine Manufacturing Hub, and it was guided by Professor Comparative and Professor Shah. at the Department of Chemical Engineering at Imperial College London. And this research was funded by the UK government's Department of Health and Social Care, and it was administered by the EPSRC. So I'm going to talk to you about vaccine production. We are coming from a chemical engineering background, so we'll talk about the production processes. And in particular, I'm going to talk about two types of modeling that we did. So characterizing and aiding the development of RNA vaccine production processes. And these two types of modeling are techno-economic modeling and modeling for quality by design. I will start with the techno-economic modeling first, and then I will close the webinar with talking about modeling for quality by design. So just giving you a brief introduction, as you are probably aware, developing a vaccine from the early research phase all the way through the phases of the clinical trial and to commercialization, it's a lengthy process. This takes an average between 8 and 14 years. It costs several hundreds of millions of dollars, can cost $1 billion or even more than $1 billion, and it has a very low success rate. So only 7% of the candidates that enter this pipeline from the R&D phase make it all the way through the approval phase. Now, this process has been accelerated in the pandemic, and that's due to two different things coming together. One of them is that we had new technologies. These are, for example, one of them is the RNA vaccine platform, which has actually been in development for about 2 decades before the pandemic. And the other thing is that because there was a pandemic, there was lots of effort and lots of resources we put into fast-tracking the development of these vaccines. And some of these regulatory timelines, or like the phase of the clinical trials, for example, have been carried out in parallel, and the regulators were on board to approve this vaccine as fast as possible, and that without cutting corners, and it's fully ensuring safety and quality of the vaccines.
Zoltan, I'm really sorry to interrupt. There is some people have difficulty with the audio, so if we can, if you can speak closer to the microphone and a little bit louder.
Okay.
Hopefully to let's try, and if there are more issues, then maybe I'll just unfortunately interrupt you again.
Sure. Feel free to interrupt. I can also change my settings on my audio settings, but I hope it's better now. So I guess I'll just continue for now. And if there's any issues, just let me know, okay?
All right. Thank you so much. And I'm sorry about that.
No worries. I'll try to sit closer to the microphone as well. All right. So I was talking about this timeline, which looks at developing the vaccine product. However, in parallel to this, there is a separate timeline which looks at developing the production process based on which these vaccines are produced, right? And that's shown on this slide, right? Now, for conventional vaccines, this was, again, a lengthy process. It took on average between 6 and 9 years to develop the production processes, all the way from a conceptual process design all the way to validation and regulatory approval of the process. As I mentioned, this took on average between 6 and 9 years, and this costed several hundreds of millions of dollars. And this timeline was somewhat in parallel with the other timeline that I showed on the previous slide. Now, with these new emerging technologies, such as the RNA vaccine platform, this timeline can be substantially reduced, and the costs can be substantially reduced as well. And this cost reduction, time reduction, can be tenfold, basically. So that's very promising. And these technologies, as I mentioned, they are platform technologies, which means that with the same base underlying technology, it is possible to produce products against different disease targets, for example. So a platform technology means that you have one base technology which enables the production of different products with the same technology. And an example for that is the RNA vaccine platform. We'll talk more about that later on a bit. So if you have these platforms, these offer very exciting possibilities to develop complementary bioprocessing modalities and computational models, and the combination of these two will be very powerful because they can together help to develop and produce vaccines faster, produce vaccines at lower cost in these flexible processes, which can be used to produce vaccines against a wide range of different diseases. And all of these together, the modeling tools can help basically improve the performance and efficiency of the production process. And if the models are used appropriately, this can also offer a lot of knowledge which can help regulatory submissions, and they can serve as a pre-qualification for the platform. And this can then ease the scale up as well of these technologies. And once these platforms are well characterized and well digitized, if you will, with computer models, then these can be tech transferred easier, and this can enable easier distributed manufacturing of these vaccines. There are also other platform technologies besides the RNA vaccine platform. In fact, in the Future Vaccine Manufacturing Hub, where I have worked previously, we have looked at 4 different platforms, but there are other ones, for example, the adenovirus vector vaccine platform. However, in this talk, I will focus mostly on the RNA vaccine platform, and I will briefly compare this also with the adenovirus vector vaccine platform. So the production of the RNA processes, of the RNA vaccines, so the RNA vaccine production process is shown on the slide. Basically, the RNA vaccine production itself starts with an enzymatic reaction. It's called the in vitro transcription reaction, and in this reaction, the RNA is synthesized based on a DNA template by an enzyme. And this enzyme is called the T7 RNA polymerase. And this enzyme basically links together the building blocks, the nucleotides. There's 4 nucleotides that build RNA into a chain, and then this chain becomes the RNA. And this chain is built by this enzyme based on the template DNA. That's how the actual production process starts. And the reaction mix that you would need for this to happen, basically, it's a well-defined reaction mix with less than 20 components. So it's a fairly simple reaction mix as far as vaccines go. It's well-defined. It's basically an enzymatic reaction, with the main components being the enzyme, the DNA template, the nucleotides, cofactors, and a couple of buffer components to maintain the pH and so on. So once this RNA is synthesized, then the next step is to digest the DNA template, and from that point onwards, the largest component in this reaction mix is the RNA itself, and then this can be separated. One of the crucial aspects with RNA is that the RNA has to be kept. So a special structure has to be added at has to be added to the 5' end of the RNA, which is basically the beginning of the RNA chain. And there's 2 main ways of adding this cap there. One is to add the cap during the synthesis. This is co-transcriptional capping. And for this, you can use cap analogs such as ARC or CleanCap, which are basically incorporated into the RNA during the synthesis. And basically, the RNA synthesis starts from elongating these caps, basically. So that's one way of capping. The second way of capping is to use capping enzymes, and this is called post-transcriptional capping because capping happens after transcription. And if you use this capping enzyme approach, first you need to synthesize the RNA, then you separate, purify the RNA, and then do the capping in a separate reaction. Pfizer and BioNTech use co-transcriptional capping, whereas Moderna is using post-transcriptional enzymatic capping. And there's several quality attributes of the vaccine product that has to be met and maintained in this production process. I'll talk about those in the second part of the talk. But basically, after the synthesis and the capping and the digestion of the DNA template, the next step is to purify out the RNA from this reaction mix. And purification can be achieved by a combination of filtration and chromatography techniques. The companies commonly use tangential flow filtration in combination with at least one chromatography technique. Now, various different types of chromatography techniques can be used here. Reverse phase chromatography seems to be a preferred one in industry at the moment. But besides that, RNA can be purified based on ion exchange chromatography, based on hydrophobic interaction chromatography, based on oligo-DT chromatography, knowing that the oligo-DT will bind to the poly-A tail of the RNA, and different types of multimodal chromatography or core bead chromatography. Once the RNA is purified, then the next step is to encapsulate it into lipid nanoparticles. Now, this encapsulation is actually the newest part of the process because all the other operations have been used extensively in the past. So tangential flow filtration and all of these chromatography techniques have been used in the past. There's nothing new about that. They have just not been applied for RNA at this scale. But the newest part of the process is the LNP encapsulation. So in this LNP encapsulation step, what happens is that the RNA is enclosed into these lipid nanoparticles. So it's basically like a liposome composed of 4 different lipid components, out of which one is the newest component. And there's also intellectual property around that component, that is the ionizable lipid. But in any case, so there's these 4 lipids which form a lipid sphere around the RNA, and the RNA is inside the lipid sphere. And this LNP is meant to stabilize the RNA and to help its delivery into our cells after injection into the tissues. So anyways, going back to the production process, we have the LNP encapsulation step. For this, there are several different procedures in place, different unit operations. This can be achieved using microfluidic mixers. It can be achieved using impingement jet mixers, for example, which operate at higher pressures. And this is a more scalable approach, or with different mixers such as vortex mixers and so on. After this LNP encapsulation step, then the next step is to, again, purify out these lipid nanoparticles which contain the RNA inside. And this purification is required because in the mixing step, when the RNA is mixed with the lipids are coming in an ethanol stream, and the RNA is coming in an aqueous phase at a low pH. It could be a citric acid buffer or an acetic acid buffer. But anyways, you would need to remove the ethanol, basically. And for that, tangential flow filtration is used. Following that, the next step is dilution and sterile filtration. And then this is the drug product that we produced, which is basically LNP, which contain lipid nanoparticles which contain RNA inside. And then this drug substance can be shipped to separate facility for fill and finish. We have built detailed models of this, starting from the mass balance and working out the equipment requirements, utility requirements, labor requirements, and all the different aspects of the process, including the facilities that will house this process. And we have built such models using a software called Superflow Designer. And this slide shows how the process looks like in the software. You can see the different parts of the process here. There are several uncertainties in this process and several potential for variability, both in terms of when you design the process and when you operate the process as well. And these are listed on this slide. So the production titles can vary, the process scale can vary, the price of the raw materials can vary as well, and the downstream purification losses can vary, and the RNA amount of those can vary as well. And we can have different field to finish approaches as well. And for each of them, we list the different ranges and baseline values, which actually are close to the realistic values based on which these vaccines are produced. And then we have carried out sensitivity analysis to see how these uncertainties impact the performance of the process. And in terms of performance, we have looked at the annual production amounts and the cost per dose, which are listed on the horizontal axis here. On the vertical axis, we listed the different uncertainties such as scale, failure rate, titer, and RNA amount per dose, and so on. And for each of them, we listed the ranges, and the zero here on the on this horizontal axis, the zero represents a baseline scenario, which is basically the center or the middle values in these ranges, and the baseline scenario is also described in this table here on the left-hand side of my screen. And then, as you can see, basically the key takeaway message here is that the RNA amount per dose has the largest impact on the annual production amount if the process is modified within these ranges of the inputs, right? And the cost per dose is most strongly impacted by the RNA amount per dose and the titer and the scale of the process as well. We have also carried out a case study where we have looked at the different vaccines that are being used or that have been developed for COVID-19, different RNA vaccines. And this study was published in end of last year, basically, so it's nearly 1 year old now. And at that point, the candidates were the Moderna vaccine, the BioNTech Pfizer vaccine, CureVac. There was a vaccine developed at Imperial College London, in development, self-amplifying RNA vaccine. And we also included a next generation of a self-amplifying RNA vaccine, which has even a lower amount of dose. And the properties of these vaccines are shown here. Now, since then, as you know, the first two vaccines have been approved by the regulators for emergency use, and then some of them, the Pfizer BioNTech one has also obtained the final approval as well. And then we looked, basically, for all of these 5 vaccines, we looked at how scalable these are and what scales are required to meet certain production targets. And on this slide, we have plots which show the scale on the x-axis. So this is the scale of the process, and it's represented by the bioreactor working volume. But when the bioreactor working volume changes, the entire process is scaled accordingly. And on the y-axis, we have the cost per dose and the annual production amount. Basically, the key takeaway message here is that as you reduce the RNA amount per dose, the cost per dose decreases dramatically, and the annual production amount increases, right? And as you can see, there's a linear relationship between scale and annual production amount, and there is this 1/x relationship between the scale and the cost per dose. Yes, and then we have also looked at how so basically what capital costs, operating costs, and what resources will be required to produce 8 billion doses of these different RNA vaccines. So in this plot, we have the RNA vaccines on the x-axis. So these are the 5 vaccines that we mentioned before, and these columns represent the capital costs, which are shown in these dark gray bars, and the operating costs shown in the light gray bars. The other bars represent different scenarios, low and high scenarios based on uncertainties in titer and other uncertainties. And these values here are representative of the baseline scenario, basically. And again, the key message here is that as you decrease the cost per dose, both the capital costs and the operating costs decrease substantially. Now, on the other plot here, on part B of this figure, we have the production scale required for producing 8 billion doses and the number of batches, production batches, required for making 8 billion doses of these 5 vaccines. And again, the key message here is that as the amount per dose decreases, the production scale requirements and the number of batches decrease substantially as well. And it's worth noting that the scale is not even the same. So the scale is 30 liter bioreactor working variant scale for the mRNA vaccines and actually smaller for the siRNA vaccines. And even with the smaller scales, you still need low number of batches to produce 8 billion doses of vaccines. So that's quite promising. And this shows how important it is to have a low amount of RNA per dose if you want to maximize productivity in the production process. Now here we have also compared RNA vaccines with another leading vaccine platform, which is the adenovirus vector vaccine platform. And for this, we use the ChAdOx vaccine platform, which is developed by Oxford University in collaboration with AstraZeneca. And I'm not going to go into a lot of detail here. You can read more about this. We have a preprint available on this, and show the link to the next slide. But basically, we looked into how the scale, titer, and amount per dose impacts the different cost components and the end activities. I think more importantly, what we talk about this next. So far, we have looked at how each of these individual inputs impact the outputs individually. However, in reality, these inputs can vary together, right? So you can have co-variation of these inputs, and this leads to a very large number of potential scenarios, and that might have different impact on the outputs, right? To capture this co-variation of these uncertainties, we have performed global sensitivity analysis based on which we have listed these inputs. So there's 6-7 inputs that we listed for each of the vaccine platforms. And then we had a certain type of distribution for each of these inputs. And then we randomly sample values from ranges of these distributions to perform this global sensitivity analysis. And we automated 10,000 production runs. And in each run, a unique combination or a unique sampling was obtained from these ranges. And then we obtained a very large number of outputs as well. And this allows us to identify which of these inputs have the highest impact on the outputs when there is this covariation of complexity in this. Now, this slide shows the SOBOR indices. So these are sensitivity indices which are obtained based on ANOVA-based decomposition of the uncertainties. But basically what they say is that the higher the SOBOR index for one of these inputs, the stronger the impact of the input is, basically. So again, this confirms that the annual production amount is most strongly influenced by the RNA amount per dose. The cost per dose is most strongly influenced by the RNA amount per dose, by the titer, and by the scale. And we have conducted an extensive study by comparing these different ranges of inputs, as I mentioned, for the adenovirus vector platform and for the two RNA platforms, for the mRNA platform and the siRNA platform. For each of them, we have the ranges listed here, the probable values, the type of distribution, and so on. We have also, in the study, we have also included field-to-finish technologies. There's several field-to-finish technologies, and out of these, the conventional filling into 10 dose or 5 dose vials was studied in more detail, because this is the technology which is actually used for making these COVID-19 vaccines, both RNA vaccines and adenovirus vector vaccines. Now, in the next slide, I'm going to show you the results from this sensitivity analysis. And here again, we have these bar charts and scatter plots. So the bar charts basically show the SOBOR indices for the different platforms, we show the SOBOR indices for each output in function of each input. And as you can see here, if we look at what impacts, for example, most the cost per dose for the doses produced per year, it's the scale, the titer, and the amount per dose, right? And this again confirms what we've seen before. And here there's a comparison between adenovirus vector vaccines and RNA vaccines. And as you can see, these bars for the amount per dose are higher for RNA vaccines than for the AAV vaccines. And this shows you the relative importance of these different inputs and how they impact the outputs. And then we have here scatter plots, which basically show the magnitude of that impact as well. So each of these dots in the scatter plots is one of the model 1 simulation. So each scatter plot contains 10,000 dots. And the shape of the scatter plot is also very informative. It can tell you how closely the input on the x-axis impacts the output on the y-axis. If the scatter plot is condensed, and if it resembles a line, that means that the input on the x-axis has a strong impact on the output. On the other hand, if the scatter plot is spread across, and it just has a large variation, that means that the input has a less strong impact on the output. Okay. And we also did this study for saRNA vaccines. And probably one of the key takeaway messages here is that the saRNA platform, so this is the self-amplifying RNA platform, can achieve low production costs and high production amounts compared to the RNA vaccine platform. And to compare better these types of production metrics, we have also plotted vial plots. So this is another way of representing the analysis to answer the question, how fast can RNA and AAV vaccines we produced. Now, these are violin plots. Again, here the length of this plot in this case represents the spread of the values. The median is shown in this wide plot, and the length is again showing the spread. The thickness or the height of the violin plot shows the distribution of the probabilities. So the thicker the plot in that region, there is a high probability to be at that value, basically. In the A part of this figure, on the top part, what we did, we compared the technologies in terms of how much time would it be required to produce 1 billion doses per production line, per facility. And the time is expressed in years. Now, these processes are conventionally implemented at different scales. So the AAV process, so this is the viral vector process, this is implemented at a 2000 bioreactor working volume scale conventionally. The RNA vaccine production process, it's implemented at a much smaller scale, something like 30 to 50 liter bioreactor working volume, right? So, and these scales are shown in this plot. But even with the scale differences, even the smaller scale RNA production processes, as you can see here, they can take less time to produce 1 billion doses. Now we have chosen this 1 billion doses as a unit because this is a sufficiently large value that from here, if you want to scale it up even to more number of doses, to higher number of doses, the scaling can be more or less linear, right? So if you want to go to 2 billion doses or 4 billion doses or 5 billion doses, or even for the global demand, this can be scaled more or less linearly. And the 1 billion doses as a unit. Now, because there were differences in the scales, then we also looking at the volumetric productivity. So basically, if you express production in terms of number of doses produced per liter per unit scale, per unit time, that gives a better picture of the productivity. And this shows us actually that the siRNA platform and the RNA platform can be about 3 to 4 orders of magnitude more productive than the adenovirus vector platform. I think I should speed up a bit. I'm running out of time. We have still a lot to cover. So I'll move on to the next slide. Here we looked at what resources are required to mass produce these vaccines. And these are again violin plots, but the key messages here are that RNA vaccine production has a lower capital cost investment requirement, but it requires a higher operating cost compared to adenovirus vector vaccine. And this is due to the fact that the facility footprint for making RNA vaccines is lower. And interestingly, as a consequence or somewhat related, adenovirus adenoviral vector vaccines have a higher fixed cost, and RNA vaccines have a lower fixed cost. That means that if you want to maintain surge capacity, RNA vaccines would be more cost effective because the fixed cost is lower. Now I'm going to switch gears now and talk about quality by design. So quality by design is a systematic approach which basically combines product and process understanding based on sound science and quality risk management in order to consistently assure the production of high quality vaccines. We have performed a case study on applying quality by design for RNA vaccines, and these are some of the quality attributes that we have identified, and the quality attributes are listed here, and these are ranked based on a risk assessment based scoring as shown here. And this is a sub list, and in fact we have a lot more of quality attributes for RNA vaccines as shown in this slide. Now, the way we apply the QbDD framework, or the quality by design framework for RNA vaccines, is to start from the patient needs. So we identify what the patient needs are. In this case, it's there's a need for a vaccine against the SARS-CoV-2 coronavirus. And then based on that, we identify the product specifications. This is the quality target product profile. And from there on, we determine the critical quality attributes of the product. And this was again determined based on the risk assessment scoring, as shown on the previous slide. And after that, an interesting step is to link these quality attributes and critical quality attributes to the production process. And to link this can be done on a unit operation basis. So for each unit operation, we can link the CQAs to the unit operation and to the parameters, to the operating parameters in that unit operation. And for this, we have designed a new ranking methodology, which takes into account both the magnitude of the impact that the parameters have on the quality attributes, the shape of the impact, and the direction of the impact. Once we have identified the relationship between the process parameters and the quality attributes, so this is linking the production process to the product, then we can also build mathematical models which describe these relationships better. And since here we are talking about RNA vaccines, and this has been applied for the RNA synthesis bioreactor, we can describe some of the quality attributes in terms of transcription rates and degradation rates. And this in turn can be defined based on enzymatic kinetics, right? So we have enzymatic kinetics for transcription and degradation, as shown on the slide, using these equations. And these two help us to obtain an effective yield, which is a quality attribute that we used in this assessment. Now, once we have this model, then we can run a large number of scenarios. And for example, slide keep changing. Here we go. So here we have a three-dimensional plot with 3 process parameters on each of these axes, and we have the effective RNA yield represented by the colors, basically. So the effective RNA yield, when it's yellow, it's low. When it's green, it's high. So you would want to be in the green space. So you would want to create the process in a space, selecting the parameter ranges so that you obtain this green area. However, in this process, there are some uncertainties, and we also incorporated an uncertainty analysis materials. But basically, in the QbDD framework, this helps us to define the design space. Now, once we have the design space, within that, we can define a subspace called the normal operating range. And the process can be then operated in this normal operating range. And that gives us flexibility to optimize the process and to control the process more effectively compared to how processes are operated and controlled currently. Right? As I mentioned, we did some uncertainty quantification, so looking at how certain it can be that we reach a certain RNA yield, a minimum yield. So the red, meaning that we're more certain, so there's high probability of reaching the yield under when we plot in function these 2 parameters. And the uncertainty was introduced by the fact that there was a 20% standard deviation in the kinetic rate constant in this model, right? And then you can operate the process in different red areas here. However, at different red areas, you have different costs of these materials. And to take the cost consideration into account as well, then we generated these other plots shown on the right-hand side of my screen here, where you have the cost of the components on this vertical axis, and you have the 2 input parameters on the other axis. And we would want to operate this process low on the cost axis and in this yellow region. So basically, this means that you can define a normal operating range around this black sphere here, right? Now, combining the RNA platform with the quality by design framework enables many things. This is a very powerful combination because both of them are disease agnostic. And this allows to incorporate prior knowledge, experimental data, process understanding, and modeling to produce a wide range of vaccines. So this is very powerful because both the RNA vaccine platform and the quality by design framework are disease agnostic, and they can help. So the quality by design framework can help both the development of RNA vaccines and can also streamline the operation of the production process. Okay, and there's different modeling techniques that can be used, but probably I'm not going to go into much detail here in the absence of time. This slide just gives you an overview, basically, of how the two modeling methodologies, the quality by design framework and the techno-economic modeling approach, can be used to accelerate vaccine development. So in the quality by design side, we have the product-specific parameters, such as the quality target product profile, the CQAs, and the CPPs are related to the product. And this diagram just shows you how the information would propagate from the product side into the process side and in between, and how this can help to improve the development of RNA vaccines in order to meet the needs of the patients. And the techno-economic modeling can also help to develop processes in a way that they will be cost-effective, they will be high throughput and fast to produce vaccines against a wide range of diseases. Now, one of my final slides is that we're also working on a supply chain model where we are looking into the whole chain of events that happen from raw material sourcing, production, and distribution, and including administration to patients. And this is a very interesting work, and we're also looking into, we're also developing a model for basically describing how the epidemics develops. And this is a team effort. Dauda, Kyonji Maria, Professor Benoit, Professor Comparative, and Professor Malaysia are all involved in this. So I'm reaching to my summary slide. So in summary, as I mentioned, RNA vaccine production is fast. It can require less upfront capital investment cost, and it has a relatively low facility footprint. The combination of the quality by design framework with the RNA vaccine platform can further accelerate the development of these vaccines and can also speed up the manufacturing of the vaccine to produce vaccines at high quality. And this can then be applied also to RNA-based therapeutics, which are being currently developed. So this can be used to target a wide range of diseases. Now, as a future work in my current role, which I'm just starting now, I'm embarking on a journey of developing and innovating these RNA production processes and to use digital tools to support this development and the operation of the final processes as well. And these digital tools and the process development, experimental work will go hand in hand and they will complement each other. So that's quite exciting, but I'm just starting this now. So hopefully I have more on this in a year's time. And as I mentioned, this work has been carried out at Imperial College London. I've listed people who contributed to this work, people from the Department of Chemical Engineering and also from the Faculty of Medicine, from Professor Rabin Shafak's group. We collaborated also with Dr. Anna Blanding, who is now at the University of British Columbia. And I would also like to thank the funding bodies that funded this work, as shown in this slide. And thank you for your attention.
Thank you very much.
Take questions.
Thank you very much, Zoltan, for this interesting presentation. We have time for a few questions. If people want to put those in the Q&A function of Zoom, which I believe you can find at the bottom of your screens. So while I give our attendees the time to type the questions in the chat, maybe I can start with one. And I just wanted to ask you, let's say, moving now hopefully towards the end of the pandemic and the end of COVID, which lessons do you think, or advancements in the space of mathematical modeling and digitalization, that can actually stay with us and sort of benefit future vaccine manufacturing, and how would that be possible?
So thank you for the question. It's very interesting. So, in my opinion, I think all the models and the tools that we developed, they are transferable to different processes as well. But more importantly, here we're talking about platform technologies, which means that these production processes can be used to produce vaccines against different diseases, right? So now you can easily reuse these tools in combination with the same platform technology to enhance vaccine production, both in terms of process development and also in terms of operating these production processes. For example, you can use the quality by design framework with all the prior knowledge coming from the product side and the process side to have all of that knowledge built into the next regulatory submission. And this would allow to have knowledge-rich regulatory submissions to in a way have a sort of a pre-qualification of the whole platform, so that you would easily know that that with this platform you can that you can use it for certain applications, basically, for example, and that would speed up the regulatory submission. Now, there's lots of potential for automating these processes as well. I'm sure, Maria, you're an expert on this, using advanced control methodologies. And this has a huge advantage as well, because these control models, for example, using model-predictive control, this can be then also be used for producing different vaccines against different diseases, or RNA therapeutics against different diseases. And this, again, almost the models are themselves a platform, in my opinion, on top of the production processes being a platform, if that makes sense. So, and the combination of the two is very powerful to basically to streamline both the development and the operation of the production processes for producing these vaccines and therapeutics in the future.
Thank you.
The question.
Yeah, absolutely. So there is a question in the chat that reads, how can you make visible the model of cost for all the parts involved to assure fair distribution and acquisition, I believe that's it?
So I understand, if it's correct, the question is how can you use these models to give more clarity in terms of what this vaccine costs, right? That's a complex question. So here we modeled only the production costs, right? And the production costs are only one part of the total costs and of the total selling price of the vaccine. So, for example, when you have a vaccine developed, you need to invest in R&D, so you have research and development costs. You have to invest in clinical trials. You have the cost of clinical trials, and there's other costs as well, right? Legal costs, marketing costs for some products. There's the cost of distribution and supply chain costs. In any way, these costs can be several billions of dollars in total, right? And the production cost is just a tiny part of it. But if you want to look at the production cost only, then, for example, you can produce a breakdown of the cost and look at, what it costs to make the vaccine. But I guess what I'm trying to say is that the production cost won't give you a full picture of the overall costs. It's only one subset of the costs, right? But we have published papers on this. We have looked at, most of this global representative here is already published, and there's breakdown of the costs, and which can basically inform how much these vaccines cost to produce. But again, selling them and selling price is much different. And in the selling price, there's also a profit margin because these companies have taken a risk and they have to cover losses from other projects as well, which have failed in the development pipelines, right? As I mentioned, these vaccines have a low success rate, so more often than not, they would fail, right? And they have to fund that in some ways, and use generally they have high profit margins for the successful products to cover the cost of the non-successful products. Okay, I guess that's my answer.
Right, thank you very much. Let's see if there are any other questions in the chat. We have time for one more question, possibly. In the meantime, just a quick one from me, maybe, on how do you assess essentially that this surge in manufacturing capacity could actually be maintained in the future, potentially using those tools as well?
Yeah, that's an important question as well. So I guess the question is, how can we have a dedicated surge capacity in the future so that we are in a better position to respond to pandemics for an unknown disease? Now, for this purpose, the platform technologies are very well suited because they offer the flexibility of making vaccines against different diseases in the future. So, but maintaining such capacity in non-outbreak times is challenging. So it would mean to have extra capacity, right? Potentially, one way of doing it is to have production facilities or factories built at a higher capacity and operate those factories at, let's say, like 70% capacity. So we have a 30% capacity buffer there in case there's an outbreak. You can quickly increase production amounts by 30%. This has the advantage that if you have this facility at the high capacity that it operates under the capacity, you would have the employees engaged and you'd have the staff engaged into running the factories. The facilities would be maintained, and you would not need to basically maintain an empty facility, which is always challenging, but you have a facility which is maintained at the lower capacity than its maximum capacity. So I think that would be a potential way, but that's, I guess it's not an easy answer, and it's not, probably it comes down to how that is funded, because if you operate at not full capacity, then you have some fixed costs, so you don't operate that efficiently, basically. But on the other hand, if you have the RNA platform, which has lower fixed costs and higher operating costs, it doesn't make a lot of difference there. So basically, you can operate effectively at a lower capacity as well, basically, because the main costs are the variable costs. So the cost of the production will be influenced by the variable costs, not the fixed cost, and which is basically independent of how much you produce to some degree, right? It gives you more flexibility. But I hope that answers the question, Maria.
Yeah, totally. And I'm wondering whether we can make room for one last question that just showed up in the chat. That says, how do you assure that the mRNA vaccine platform can be used for other pandemics at the right cost, at the right time, when the next pandemic comes around, basically?
Well, that's again, not an easy question. How do you assure? Well, one way would be to have RNA production facilities distributed around the world, right? So then we would have what we call distributed manufacturing, which would have the advantage of responding quickly to different demands, to local demands around the globe, right? But that comes with different challenges. assuring the quality of the vaccines to reduce all of these different sides is a challenge, right? So how will you assure the quality? Yeah, that's a challenge basically. The other challenge is that you will need to supply these raw materials to all of these different sites. And right now, the supply chain of the raw materials is still kind of new for RNA vaccines because this is a new technology and it's still, yeah, so there's still room for maturation and improvement there. But hopefully over time, that could be a solution. But there's still challenges at the moment with that. But having this distributed manufacturing, and as I mentioned, operating the facilities below capacity to make vaccines for other diseases such as routine child's immunizations or vaccines for trials at subcapacity, that would be a way of maintaining and keeping those facilities in a way active. And then if there's a pandemic, those facilities can be quickly, used with the spare capacity, with the buffer capacity to make vaccines against different outbreaks. So yeah, that's a solution that I'm not, yeah, there's, I think this is still very much an open discussion, and I don't think there's a clear answer at the moment. And I'm also very interested in this topic, but yeah, that's at least my view on it. So I hope that helps.
Thank you very much, Zoltan. I'm conscious of the time, so I guess I'd like to thank you for sharing with us your exciting work and views on how these digital tools can actually ramp up manufacturing and how can we actually establish the learnings from COVID to advance the vaccine space in the future. I know there are some requests from people who would like access to the recording. I'm pretty sure that IMSE can make this available, but you can double check with an IMSE representative, or maybe we can have this answer right now in the chat. In the meantime, and before closing this event and thanking you all for your attendance, I'd like to remind you of our next webinar, which is on the 21st of October at 2, with title Forecasting COVID-19 Uptake in the UK and Monitoring Attitudes to COVID-19 Vaccines, presented by Dr. Alexander de Figueiredo. And also keep an eye for the annual lecture, as I mentioned at the beginning of this webinar. I just see a comment now in the chat that the recording of today's talk will be on YouTube. So keep an eye for that too. And with that, I would like to thank you all for your attention, and Zoltan for sharing your work with us today. Thank you very much. Have a good rest of the day.
My pleasure. Thank you.